We are the U Calgary Biomod 2026 team, and we are presenting Aptonix. Me and our team of 8 wonderful individuals across biomedical engineering and biological sciences are all creating a modular biosensor for Alzheimer's disease. Our team is passionate about health research and innovation as a whole, and it's driven us to tackle a lack of preventative healthcare for degenerative diseases, specifically Alzheimer's. The reason for this is because 1 in 13 adults over 65 develop Alzheimer's, and for those over 85, it's a 1 in 3 chance of developing Alzheimer's, which creates a ton of issues when it comes to brain and things like that, especially considering that when we get to the end stage of Alzheimer's, it's not curable at all. But we have to look at some biomarkers as to how this kind of happens. Beta amyloid plaque builds up around the neurons, and tau protein tangles in between the neurons, which can cause issues with synaptic issues as well as brain shrinkage as a whole, and just cause the death of multiple neurons. And at the early stage, it can cause memory loss, difficulty learning, and mood changes. But at the severe end, you could loss of speech, physical decline, and having to be fully depend on someone else. So why is it important to solve? Well, the biggest thing is that these are part of the worldwide UN's health goals, one being good health and reducing inequalities. And the main issue for this is because current testing is very, very financially intensive. That can be very expensive, have long wait times, and so on. And it's really hard to make tests that are very consistent.
Current solutions for Alzheimer's early detection: neuropsychological testing, cerebrospinal fluid analysis, antibody-based blood testing, PET and MRI scans. All of these are viable in their own ways, but there is a missing universal gold standard for Alzheimer's early detection. A key con against each of these methods, beside the spinal tap and the antibody-based blood tests, is the dependence on late-stage pathologies. That is, once they have a true positive signal, the disease is often progressed into moderate to severe stages.
So this is where we propose our solution. We propose a DNA-based biosensor that detects proteins characterized by Alzheimer's disease directly from a patient's blood sample. We use aptamers, which are short-stranded DNA that folds into specific shapes and allows them to bind very precisely to our target protein, and acts as basically a highly selective receptor. We also have a synthetic dye, methylene blue, which will be used to convert the protein event into an electrical signal. And then we have a toehold mediated strand displacement, which is basically just a mechanism that allows us to make our sensor more modular, so it won't be just stuck to the aptamer we chose, but can be used with other aptamers as well. And finally, we have a dendrimer, which is a highly branched set of molecules. It doesn't have to be organic, but it's similar to this tree structure, and basically it provides a structural scaffold which holds many aptamers together in a small area, which increases signal strength and detection sensitivity. Finally, the entire system is connected to an electrode, which allows us to actually detect the signal by measuring the current.
Our solution allows for earlier detection of Alzheimer's disease because the high specificity of our receptor allows it to detect Alzheimer's associated proteins before extensive plaque formation and irreversible neuronal damage. Converting the protein binding event into an electrical signal provides instantaneous quantitative results. The modularity of our sensor is also an environmental advantage. Instead of replacing the entire biosensor, only a short DNA receptor needs to be swapped between screening sessions, reducing the waste material. Our biosensor also consumes significantly less energy than a PET scan, contributing to a smaller environmental footprint. Our biosensor is also ethical and cheap. Since its key component can be chemically synthesized in a laboratory, this eliminates the need for animal-derived antibodies and supports scalable, large-scale manufacturing of the biosensor. The test is minimally invasive, requiring only a simple blood sample and avoiding radioactive tracers or x-rays. This makes it easy to administer and integrate into routine health checkups in hospitals and clinics, increasing accessibility and enabling broader early screening across the population.
So what is our plan? Our plan is we have two different teams. One team is a wet lab where we prepare the aptamer receptor and prepare the dendrimer scaffold, and basically that's where the actual building is going to happen and actual validation. Before that is where the dry lab comes in. They make the modular molecular interactions and help us optimize this biosensor for binding, and just simulates the whole thing so that we can make more optimized decisions in the wet lab. And our whole timeline going from March to August is basically going from a system modeling and literature review to understand what we want to do. And May and June is where the actual experimentation will take place, where we look at from the dry lab, look at how the actual signal will work, and for the wet lab, looking at how the aptamer will bind. And finally, from July to August, we'll have the full assembly, the signal testing, and the final validation of our fully selected biosensor.
So to wrap it up, Alzheimer's progresses silently for many years, while today's best detection tools are often expensive, invasive, or hard to access. Our solution is a reconfigurable electrochemical aptamer-based biosensor that can translate early biomarker bindings in the blood into a clear electrical signal we can read, both quickly and affordably. The big advantage with our solution is that we can reuse the same sensor hardware and swap the DNA recognition module using TMSD, which lowers the cost and waste while also improving accessibility.
Now, obviously, while we have the sensor, there's going to be a lot of hurdles that come across the way. One of the biggest ones being funding, where we need funding to get all the reagents and aptamers that we need in order to create this actual sensor. And second off, we need a lot of mentorship because, as we said, our dry lab can help us optimize, but what helps us more is having experience because at the end of the day, that is what will get us our foot in the door. So, thank you for helping us, and let's help Aptonix bring the preclinical Alzheimer's screening closer to accessibility for everybody.